Okay. Excellent. Okay. First of all, thank you very much for the invitation to present today some of the work we have been doing on adaptation in wild sunflowers and its genetic basis. I should apologize beforehand. I am a bit under the weather, so I'm a bit sluggish than I would usually be, and the presentation might be less snappy than I want it to be. But that's what we have. So yeah, I'll just dive right into it. So maybe if I manage to change slides, yes. Yeah, so as you know, both plants and animals have evolved a remarkable variety of strategies and approaches to survive and adapt to different environments. And this results in the incredible diversity that we see in nature between species, but also often within species. So you can have different populations, which are also often called ecotypes, within the same species that have very different characteristics that help them adapt to the particular environment where they grow. And really the focus of my work is trying to understand what are the, try to get as comprehensive an understanding of this adaptation as possible, which means understanding what are the genes that control this phenotypic variation in natural populations, how this variation is important for allowing these plants to interact with their environment and adapt to it. And then ideally also trying to understand how this knowledge can be used and translated into crop improvement. As part of this kind of multidisciplinary approach to adaptation, I also sometimes happen to work with metabolites, with plant metabolites, and in particular with pigments. So my talk today will be on a couple of different stories I've been working on that have to do with pigments and the role in adaptation. So the first part of the talk will be about the paper that Lars mentioned before. So the UV patterns and inflorescences of wild sunflowers and their somewhat unexpected role in adaptation. And then in the second part of the talk, I'll briefly talk about a project on seed camouflage in young sunflowers. And I should preface all this by saying that by training, I am a geneticist, molecular biologist, maybe evolutionary biologist. And there's going to be a bunch of that stuff in this presentation. I am definitely not a biochemist. So I apologize beforehand if I say something that is grossly wrong or incorrect. Please be patient with me. But also I am very excited to present in front of this audience because any suggestion or comment or criticisms you might have will be really very welcome because you know much more about this than I do. So first of all, it's probably good to start with a brief introduction about wild sunflowers and why we study them. Wild sunflowers are obviously, so they are the wild relatives of cultivated sunflowers. They're very similar to cultivated sunflower in that they look like sunflowers, but instead of having like the sunflowers you see in a field, just a single inflorescence, like a big thick stem and no branches. Wild sunflowers are generally very branched. They have many smaller flowers. And the reason why they are interesting from adaptation and evolutionary biology and ecology perspective is that there's many different species of wild sunflowers. They're all native to North America, and they really grow everywhere in North America. So they are adapted to grow also in fairly extreme environments, sand dunes, salt marshes, very dry places, flooded plains. So there's a lot of raw material for the study of adaptation that one can work on. The other advantage of wild sunflowers is working with wild sunflowers that partially because they are related to cultivated sunflower, there's really a lot of genetic and genomic resources for this species. So we have now, I think, more than a couple of dozen reference assemblies for different species of, like for cultivated sunflower or different species of wild sunflowers. We have resequencing data for thousands accessions of wild sunflowers and cultivated sunflower, and there are like large collections that you can access and get seeds from. So that's really a lot of material. And so, as I said, the focus on my research is on adaptation, and also this talk is going to be about color, and that means in plants, color is usually most closely associated with flower color, and flower color is really a quintessentially adaptive trait. The coevolution between flowering plants and their pollinator results in this explosion of diversity in flower colors and patterns of colors, but also shapes and scents and other things that you can see in flowering plants. And again, this diversity is mostly between different species, but often also you can see variation within a single species. So you can have different different ecotypes, different population of the same species that have flowers that look very different in terms of flower color and flower patterns. But that does not seem to be the case of sunflower at the first glance. And indeed, all wild sunflowers have ligos, which are these modified petals of the outside word of the inflorescence. So the sunflower flower is an inflorescence, so it's a ton of small flowers that will each produce one fruit, one seed, and then the outer word is modified petals that are called ligos. All the ligos of wild sunflowers are uniformly yellow to the human eye, but if you were a bee, things would look quite different. So these are the same two inflorescences, but seen in the UV. So this is a UV picture. And then you can see a bee and other insects and animals that are able to see in the UV would see that sunflowers actually accumulate UV absorbing pigment. So the dark means UV absorbing at the base of the ligos and across the whole inflorescence. This forms this UV bullseye pattern that is known, like their visible counterparts, to be important in pollinator attraction. So pollinators like this, like they are attracted, they increase the visibility of the flowers, and so they increase pollination rates. The other thing you can see from this slide is that you have also a lot of diversity in terms of the size of these UV patterns, and these are both plants from the same species, which is Helianthus, which is the closest wild relative to cultivated sunflower. And when we look in a larger collection, so we grew and phenotyped for UV patterns about 2,000 individuals from a few different species, we found really a lot of diversity. And especially for Helianthus. So this is a map of the distribution of the population that we assayed, and hopefully this is like you can understand it easily. So when the black dot is larger, means the UV patterns are larger. When it's smaller, means that they're smaller. So you can see that there is a lot of diversity across populations, and really you have everything from plants that have ligos that are completely UV reflecting to ligos that are completely UV absorbing. So this was very exciting. There's a lot of variation, and two questions immediately came to mind. The first is, how do you get this variation? So what are the genes that control these patterns? What is the pigment that is absorbing the UV? And the other question is why you would have this variation to start with. Like, why do you need, in different parts of North America, you might need UV patterns that are larger or smaller? Answering the first question was relatively straightforward, because as part of a previous project, we had already resequenced several hundreds of the plants that we phenotyped for UV patterns, so we could just do a GWAS. And for Helianthus, we found that there is basically one locus that explains the vast majority of the diversity that we see at the beginning of chromosome 15. And so here, and the SNPs that have the strongest association with the size of these UV bullseyes falls in the promoter region of a gene that is called HaMYB111. So what does HaMYB111 do? It's the sunflower homologue of the Arabidopsis MYB111, which is a member of a small family of transcription factors that are also called production of flavonol glycosides, or PFGs. And as the name implies, these are important in regulating the expression of biosynthetic enzymes for the synthesis of flavonol glycosides. Again, you probably know this much better than me, but flavonol glycosides are a family of flavonoids. They have been involved in a number of responses to stresses, cold, UV, drought, also biotic stresses. But perhaps more importantly for this presentation, at least at this point, is the fact that they are also known to be the UV absorbing pigment in the flowers of several plant species. So they make for a very good candidate for UV pigment in sunflower as well. And that's indeed the case. So when we did extraction from ligos that had very large UV patterns or very small UV pattern, we found that the UV absorbing pigments in the extracts from ligos with very large UV patterns, this is the gray phrase here. So they're all flavonol glycosides. And then if you look in plants that have very small UV patterns, these same flavonol glycosides are, this is the yellow trace, are absent or almost completely absent. And that's true both when you compare ligos with large or small UV patterns, but also if you take a ligo that has intermediate UV patterns, so the bottom is UV absorbing and the top is UV reflecting. If you check at the bottom, you have accumulation of flavonol glycosides, and in the tip instead, you have no flavonol glycosides, meaning that it's very likely that so flavonol glycosides are the UV absorbing pigment in sunflower ligos. And we could also confirm that the sunflower gene has the same function as the Arabidopsis gene through heterologous complementation. So when we check in Arabidopsis, we found that MYB111, so the closest homologue to the sunflower gene, is expressed very highly in petals of Arabidopsis. And when we check, somewhat to our surprise, we found that sunflower and Arabidopsis petal actually absorb a bunch of flavonol glycosides, and they are highly UV absorbing. And the reason why I say this is a bit surprising is because, as I mentioned before, UV patterns in flowers are known to be important for attraction of pollinators. But Arabidopsis is like 95% plus selfing, so it doesn't really need to attract any pollinator. So it's not, it wasn't really clear at this point why there would be UV patterns in flowers of Arabidopsis anyway. Nonetheless, we check the mutant MYB111, and MYB111 mutant doesn't accumulate almost any flavonol glycoside in the petals, and the petals are UV reflecting. And then when we take the coding sequence from sunflower, put it into Arabidopsis, we can complement the mutant, and this is regardless of which promoter we use. So we use either 35S, so constitutive promoter, or we use the endogenous Arabidopsis MYB111 promoter. In both cases, you could have you recover accumulation of flavonol glycosides and UV absorbance in the petals, meaning that really, as I said before, the sunflower MYB111 does the same thing as the Arabidopsis MYB111, which is control the accumulation of flavonol glycosides. The other thing you can see from this slide is that we use two different versions of the sunflower MYB111 gene, one from plants that have very large and one from plants that have very small UV patterns, and both of them are able to complement the mutant. So, and this suggests that the variation, the difference between these two alleles is not between, is not in the way, sorry, it's not at the protein level, so it's not in the activity of the gene itself, but it's probably in the places where or when it's expressed. So where is MYB111 expressed in sunflower? So these are data from RNA-seq, so published previously in cultivated sunflower, but we can see that, as is the case in Arabidopsis, MYB111 in sunflower is expressed very highly in legals. And when we look more carefully, we found that actually, so we took legals, and check expression at the base and the tip. We found that MYB111 is expressed very highly at the base of the leaves where the flavonol glycoside accumulate, but not at much lower levels at the tip of the leaves of the legals. And that happens only though when we check in very in developing legals, so very small legals. They still haven't turned yellow, so they're just starting to develop color patterning, and they're starting to accumulate UV pigments. MYB111 is almost not expressed at all in mature legals, and this is consistent with this transcription factor being important in establishing these patterns of UV pigmentation early on during the development of the legal. And then lastly, we check also a bunch of different accessions of sunflowers. So these are plants that have been collected across all of North America, and we just these are 46 different genotypes. They have been selected for being either with very large or very small UV bull size, and we found that in developing legals of plants that have large UV bull size, MYB111 is consistently and significantly expressed at higher level, again suggesting that differences in expression patterns are probably what drives the difference. MYB111 is probably what drives the difference in UV bull size between these plants. And this is consistent also with the fact that, the fact that the difference in expression more than a difference in function is what drives this diversity is consistent with what I showed you in the beginning, that the strongest association in the GWAS is not in the gene itself, but in the promoter region. And we actually found that there is a lot of structural variation in the promoter region that is probably causal for this differences in expression. So this answers the first question. So how do you have this variation? It's due to transcription, so cis-regulatory variation at MYB111 that affects the accumulation of flavonol glycoside in the legals. And the other question is why you need this variation to start with. As I mentioned before, it is known that ultraviolet bullseye floral patterns are important for pollinator attraction. And so we wanted to check if by chance also pollinators are able to distinguish different sizes of UV bullseye. So if they care if you have large or small UV bullseyes. So we had a field with about 1,500 different wild Helianthus annos. These are from about 100 population from across the range, so they cover most of the diversity that we have in this species. We selected 83 plants that had that flower at the same time and the same number of flowers, and that were divided into 3 categories. So with very small UV bullseye, very large UV bullseyes, or intermediate ones. And then we just sat there for a few days and look at the bees and the bumblebees and see who was visiting which flower. And we found that indeed pollinators do have a preference. They tend to prefer some flowers that have intermediate UV bullseyes, and they really don't like sunflowers that have very small UV bulb size. So that's a nice result, and it's consistent with what has been seen also in other experiments. People have been also artificially manipulating UV bulb size and finding similar things, but it doesn't really answer the question, right? So if you have one pattern, so intermediate patterns are better than the others at attracting pollinators. Why do you need to have the other patterns at all? One possibility is, of course, that preferences of pollinators are not uniform across the range. So maybe different pollinator assemblages in some parts of the range have different preferences. That's possible, but it's unlikely because sunflower is really pollinated by many different insects. And also all the major pollinators of sunflower are ubiquitous across North America. So it seems unlikely that they would agree to have different preferences in different parts of the range. That doesn't mean that pollinators don't have a role in explaining this diversity, but it might be, so it kind of motivated us to look for other possible explanation. And the fact that there is this fairly clear geographic distribution kind of suggested that maybe environmental climatic factor might be a driver of this diversity, which is also consistent with what I mentioned before, the fact that flavonoglycosides are known to be associated with responses to different abiotic factors. So we look at a bunch of different climate variables, UV, radiation, temperature, and so on. For the sake of time, I'll just skip to the winner, so the one that we found makes more sense, and which is relative humidity. So relative humidity has the strongest association with the size of this UV bulb size across the range of wild Helianthus annus of all the couple of dozen environmental variables we checked. And it's so the correlation is such that you have larger UV bulb size, so more flavonoglycosides when you have lower humidity, so in drier places. And it is known that lower relative humidity tends to increase transpiration, so increase water loss. And it is known that flowers, especially showy flowers like the one that you have in sunflowers, are an important source of transpiration for plants. The other thing that makes sense is that it is known that flavonoglycosides have a role in regulating transpiration, regulating water loss, and promoting drought resistance. So both in Arabidopsis and corn, if you have transgenic plants that accumulate larger amounts of flavonoglycosides, they do much better when you don't give them water. So here in Arabidopsis, you see wild-type Columbia after 14 days, like very sad plants, while plants overexpressing another of the PFG, so the same family of transcription factor as HaMYB111, they accumulate a lot of flavonoglycosides and they're still doing fine after 14 days after water. And the same is true for maize. So we wonder if that might be the case also for sunflowers. So if that's a role of flavonoglycoside in affecting water loss. So we did a silly experiment. We took 16 different sunflower lines, again, trying to maximize geographic diversity. These were either plants that have very large or very small UV patterns, plucked out the legals and put them to dry in the lab and check at regular interval how quickly they would lose water. And we found that plants that accumulate, that legals that have larger UV patterns, so they have more flavonoglycosides, they lose water at a significantly slower rate than plants that have instead a small UV pattern, so they have little amount of flavonoglycosides, suggesting that this variation in flavonoglycosides and UV patterns is probably important also for helping plants regulate transpiration and possibly confer some drought resistance in drier places. So the answer to the second question, why there is such variation, this is due to probably a combination of different of the effect of flavonoglycoside accumulation on different variables, so pollinator attraction, transpiration, and so resistance to possibly drought. And both of these traits are, of course, very important for adaptation in wild sunflowers, but they're also potentially important for crops. So sunflower, although cultivated sunflower is a selfer, it is severely pollination limited, so you need to have pollinators to have sufficient yield. And also, of course, drought is one of the main limiting factor for sunflower production across most of his cultivated range. So this concludes the first part, and in this part I have shown you, not that there was any need because everybody knew it already, but I've shown you that we have seen that one of the main function of color in plants and animals alike is to broadcast oneself, so to either attract pollinators or potential mates, or to warn off potential attackers. There is another important role that color and pigmentation can have in plants and animals, which is disguise or deception, and this includes camouflage, which is the ability to avoid detection by blending in with the surrounding. And camouflage is a very well studied, very classic example of adaptation in animals. There's like if you just Google that, and you'll for sure know many examples, many cases of melanoids, for example, where you have different morphs that are adapted so that they can blend in with different parts of the environment. So very well studied in some cases. Also, the genetic basis of this diversity has been identified. It is usually, like in many cases, the MC1R gene has been involved for both, for example, for mice and for lizard pigmentation diversity. But really, so very well studied in animals, very little study in plants. Plants are usually more of the backgrounds that animals hide against. Which brings me to Helianthus anomalus. So this is another species of wild sunflower. It has a much smaller range than Helianthus annus, which is the plant I mostly talk about in the first part of the talk. So you have here Helianthus annus in gray, and instead anomalus only grows in Utah and northern Arizona. And it grows on a specific niche, so it only grows on sand dunes. And it's really quite spectacular. So these are really fairly big sand dunes. There's only some shrubs that grow there, and then you have these big sunflowers with hundreds of flowers, 2 meters tall. So it's really quite impressive. And of course, Utah and Arizona are famous for their colorful rock formation. So you have orange, red, tan, everything in between. Like mountains, you can see them in Western movies, and that's the case also for sand dunes. So you have sand dunes that are orange, sand dunes that are light tan, and you have Helianthus annus growing on both of these types of sand dunes. And the really cool thing is that the color of the seeds of these plants matches the color of the sand the plants grow on. So plants growing on orange sand dunes will have orange seeds, and plants growing on tan sand dunes will have tan seeds. And so this is really like a very nice system to doing studies on adaptation, because working with seeds is much easier. It's very amenable to ecological field experiments, but also you have all the genetic and genomic resources for sunflowers, so you can hopefully also get down to the genetic basis of this. So there were really 4 questions that we wanted to answer on this system. First of all is that, so we have like this couple of few observation of concordance between seed color and sand color. We wanted to make sure that's really a general phenomenon in this species. If that's the case, we want also to know if it's camouflage, so if it really helps prevent predation of these seeds. And then there are also more functional questions. What makes these seeds color? So what's the pigment, and what are the genes that control this variation? So to answer, like for the first part in particular, but for the whole talk, it's probably useful to go through how you get colors in seeds of Helianthus anomalus. So this is a section of the seed. Actually, I call them seeds just because it's easier, but these are fruit, of course. So the seed is the kernel inside the sunflower fruit. And then on the outside, you have the hull, which is all maternal tissue. And there are 3 layers to the hull. The first layer is the phytomelanin layer, which is always present. This is like dark gray black. On top of that, you have the hypodermis, which is white and can be present or absent. And then the outermost layer is the epidermis, which in Helianthus anomalus can be transparent or orange. And the combination of these 3 colors is really what defines the color of the seeds, right? So if you have an orange epidermis and the hypodermis is present, you see the orange. If the epidermis is transparent, you see through to the hypodermis. So you have white seeds. And then regardless of what's the color of the epidermis, if the hypodermis is missing, you see through to the phytomelanin layer. So you have these black dots. And this means also, like, obviously it's a fairly complicated way of getting to color, and this reflects also in the complexity of the phenotype. Unfortunately, seed color in Helianthus anomalus is not a binary trait, so you don't only have tan and orange seeds, but you have all kind of possible intermediate colors, and you also have different patterns. So you have stripes and spots and dots and all kinds of things, which complicates. We'll see later, we complicates the genetics, but also complicates a bit the phenotyping. So you need to find the best way to really assess the color that you're looking at in these seeds. So there's a different way of doing that. The simplest way is to just, since we are interested in this variation between tan and orange, just check the proportion of red in these seeds. So we scanned thousands of these seeds, analyze their color profile and analyze the color profile of sand, and then basically just measure RGB and then compare the proportion of red to the total RGB in seeds and sand. And that works surprisingly well. So this is a correlation between proportion of red in seeds and proportion of red in sand. And you can see that in different populations, you really have like an almost perfect correlation. So it really looks like it's a widespread phenomenon, this concordance between seed color and sand color. You can also do more appropriate, more advanced analysis. So this is a segmentation analysis. So you basically just divide an image in different color bins and see what is the proportion of that image that falls into different bins. And again, you see that plants that grow on tan sands have like much higher proportion of tan colors, and seeds, plants that grow on orange sand tend to have seeds that have a much higher proportion of orange and brown colors. So the answer to the first question is yes. There is really a very good match between seed and sand color. And I should mention that this seed color is genetically encoded, right? So if you take seeds from this population, grow them like in a greenhouse on brown soil, they still make tan and orange seeds. So it's not something that they suck up from the sand. It's really something that the plant produces. The next question is this camouflage? And does this make any difference in terms of predation? To answer that question, over a couple of years, we did a bunch of field experiments in Utah. We selected two populations that have different sand colors. So the Little Sahara recreation area has very light tan sand. The San Rafael Desert, more southern in Utah, has orange to deep orange sand dunes. And we did some very basic experiments. We took a focal plant and then in front of it, a focal sunflower plant. So these are how they look like more in the wild. In front of this plant, we put 5 orange and 5 tan seeds before dawn. So we would go there out in the desert before dawn, put these seeds, and then during the day, basically check which seeds were eaten and at which speed. And we did this over 24 hours, so also during the night. And then we did another set of experiments. We're also interested in understanding who is eating these seeds. So we did another set of experiments with exclusion cages. And this basically allows you to separate the predation from larger predators, which are birds or mammals, so rodents, or predation by ants, or just the wind blowing away your seeds. So a lot of data came out of this. These are very noisy experiments, so we did a bunch of replicates. And I'm trying to summarize most of these results in this not very straightforward plot, but I'll drive you through it. So here on the x-axis, you have different conditions that we tested. And for now, the main thing that you need to notice is that, so on the left side, here you have experiments that were done in the San Rafael Desert, so on orange sand. And on the right, you have experiments that were done in Little Sahara, so on tan sand. On the y-axis, you have a predation index. And when you have positive values, mean that more, like in that particular setup, more orange seeds were eaten. If you have negative values, means that more tan seeds were eaten. So we did this experiment, analyzed the data, and what you can see is that when you have no cages, so when all kinds of predators could get to the seeds, you have a clear effect of seed color. So you have that on orange sand, more tan seeds were eaten, and this goes away when you put seed cages, you put exclusion cages. So that means that whoever is doing the eating is either birds or mammals or rodents. And actually, we know that this predation happens only during the day, and birds are the only. So birds are active during the day, rodents are active during the night. So we know that this difference in predation is due to the effect of color on bird predation. What you have on this other side of this plot, so the initial set of experiments we did was with seeds that were propagated in the greenhouse, but they came from plants that were either from populations that were adapted to growing on the orange sand or populations that were adapted to grow on the tan sands. And these populations are about a couple of 100 kilometers away. So they are like very divergent. And we think that when we are doing these experiments, we are comparing differences in seed color, but maybe there is something else that is different between this population, and one seed tastes like chocolate and the other one tastes like wet socks. So that maybe we think we are seeing an effect of color, but it's something else. To try to overcome this limitation, we generated two populations between the plants that have between plants that had orange and 10 seeds, and then in the F2 progeny, we selected seeds that were either orange or 10. And so in this way, we are hoping that basically in between the two pools of seeds, everything else will be the same on average, because all other traits should segregate independently. And these two pools should only be different because of seed color. So we repeated these experiments, and then again, we see the same thing. So on orange sand, more 10 seeds are eaten outside the exclusion cages. And this is what we see on orange sand. On 10 sands, we see the opposite effect. So on 10 sands, you have more orange seeds that are eaten. So altogether, these experiments show that there is really camouflage, and this is due to the effect that color has on bird predation, which makes sense because birds are by far the most visual of the predators in the area. So this is the part that work beautifully. As the more I go down, the more things get a bit messier. So the next question was, what is the pigment? So why you have orange and 10 seeds? Took a while to figure out how to best extract the color out of them. Turns out that you mostly have to make a tea. So if you leave the seeds in water with some methanol for a few days, warm, you will get these nice extracts. When we went and checked what is in the extracts, turns out there is a bunch of things. So it was impossible to really figure out what of all this was the pigment. To try to simplify this, we did a developmental series. So we crossed plants that are orange and 10 seeds, and then so we took plants that have produced orange seeds and 10 seeds, and collected hulls every 5 days after pollination, and did extractions out of these hulls. They all look brown because sunflower seeds, when they're wet, they all look brown. But then if you would dry them, you would see that these actually appear, they look orange, and then this one look tan. And then you can see, so we did this extraction, and at about 10 days, you start to have accumulation of the orange pigment, and then you have much stronger accumulation at 15 days. So we look at these extracts, and we found for metabolites that follow the same kinetics, and we found that there are a couple of chlorogenic acids that follow that very well. Chlorogenic acid, it's a very abundant compound in sunflower, especially in sunflower seeds. And actually, we knew already before that orange seed accumulate higher level of chlorogenic acids. We just didn't really thought that was the pigment, because my understanding was that chlorogenic acid on its own is colorless, and then when it oxidizes and complexes with proteins, it becomes green, which is why it's called chlorogenic. So it produces green. So that's what happened when you bake your cookies with sunflower butter. They will turn green. Turns out though that that's not the only color that you can get out of chlorogenic acid, and it really depends on which amino acid it reacts with, right? So different amino acid in combination with chlorogenic acid will give different colors. The one thing that really struck me was that oxidized chlorogenic acid on its own has actually this deep orange color that is very similar to what we see in Helianthus anomalus seed extracts. So this is not definitive evidence, but I mean, it's very suggestive that probably the color of the seeds is due to accumulation of chlorogenic acid. And then the last thing we wanted to do was to see if we could understand what are the genes, what are the genetic pathways that are controlling this diversity between populations. This is a slide that summarizes a bunch of work. So we did a QTL mapping for population, 1,200 individuals. We genotyped them all with GBS. And what turns out, perhaps not surprisingly, given how variable the color and color patterns are, is that the genetic regulation of seed color is also very complex. So basically, there's QTLs in most of the sunflower chromosomes. Sometimes you have the same QTL popping up in different populations, but that's not always the case. And they tend to be fairly small size QTL. So at tops, I think 25, 30, they explain 20 to 30% of the diversity that we see. So yeah, the answer to the last question, the genetic bases are complicated. That's not a very satisfactory answer. So we are doing more things to try to look at that. So we have been doing RNA-seq analysis of the developmental series I showed before, where we did the extraction, the pigment extractions. We're also doing admixture mapping in wild populations that are segregating for seed color. We have assembled the genome of anomalus, so we hope that if you put everything together, we might be able to get some more information out of this. With this, I conclude my presentation. This is like a lot of work that has been done in collaboration with a number of very talented, very generous people that have been spending weeks climbing up and down dunes looking for tiny seeds. And also I thank you all for your attention, and I'm happy to take any question that you might have.
Thank you very much, Marco. That was a great talk.